So, hello all. We are going to start our class in next few minutes. Okay. So we are going to start today's session. So a very good evening to all of you. So I'm Apurva Soni and we'll be studying about the topics that have been covered in the week 9 in this today's lecture. Okay. So in the last week, we have studied about development of axis formation in Drosophila. So we have covered in the last 2 weeks classes, last two classes, we were studying about Drosophila axis development, where we have studied about the uh, dro- dorsal and ventral polarity and anterior and posterior axis formation, how it takes place, what are the genes that are involved, and everything regarding it. So now we'll switch for few classes. We'll study about the plant developmental biology. So when we talk about developmental biology can be animal specific, it can also be plant specific. So we are now going to study in brief about the developmental process that occurs in plant. And whenever we talk about the development process, we are specifically trying to talk about the events that occurs after fertilization. Okay, so after fertilization, what are the events that takes place and how the fertilization of the male and the female sporophytes takes place, which ultimately leads to the development of a plant. Now some important points before starting the discussions that I think we should keep in mind are um, mm, hmm. So whenever we are studying about plant development, there are certain points that we should always keep in mind. Okay, what are those points? Important points. First is that plant cell do not migrate. Unlike animals that can migrate from one place to another, plant cells they do not migrate from one place to another. Second is plants have sporic meiosis rather than gametic meiosis. That means plants do not have gametes. They have a different kind of meiosis process, which is known as a sporic meiosis because they have sporozoites. Okay, then the life cycle of most plants alternate between diploid and haploid multicellular stages. Now, this is an important point to remember. Why? Because unlike in animals where we are studying, so when after fertilization, it leads to the formation of a diploid zygote. But here, you can have a, yeah, you'll see here, you see this picture. So here you can see that there can be haploid generation and diploid generation. Both haploid and diploid generations are present in the case of plants, unlike as in animals. Then plant germ cells are not set aside early in development. Okay. Then plant undergo extended morphogenesis. Now this is again a very important point. So when we talk about that plants undergo extended morphogenesis, what does it mean? It means that, for example, in humans, so after a child is born, the morphogenesis in terms of organ, new organ formation doesn't take place. So whatever organs are formed in the when the child is born, those organs are only there. There are no new organ formation. For example, if you're born with 5 fingers, you'll have 5 fingers. You won't develop sixth finger after your after a baby is born, after you're born. So, but in the case of plants, it is not the case. Even a fully mature plant can go to increase its height, or to can it increases its stems, its leaves, its branching. So the morphology is not the same of a plant. It can undergo extended morphogenesis, whereas in the case of animals, the morphology is the same. Morphology in the means that whatever organs, whatever features a particular animal has, those features are retained throughout the lifespan of a healthy organism. Okay, then the next point is plants have tremendous developmental plasticity. What does it means? Again, that if a particular for example, just let us consider humans. Okay, now if you've cut a finger of a human, the finger cannot regrow again. But when it comes to plants, that finger can I mean the if you cut a stem, or if you pick flowers or leaves, again leaves will come at that particular position, again flowers will come at that particular position, again that stem will occur in some of the other places. So that means they have developmental plasticity, which is not seen in the case of animals. Then multicellularity evolved independently in plants and animals. So these are some of the points which differentiates the developmental process between plants and animals. Okay, I'm repeating it again. First is plant cells do not migrate. Plants have sporic meiosis rather than gametic meiosis. The life cycle of most plants alternate between diploid and haploid multicellular stages. Plant germ cells are not set aside early in development. Then plant undergo extended morphogenesis. Plants have tremendous developmental plasticity, and multicellularity evolved independently in plants and animals. And now we'll just see this, if you can see this figure. So plants have an haplodiplontic life cycles. What does it mean? What is the meaning of haplodiplontic life cycles? That means that their life cycles between a mitotic phase and a meiotic phase. That means they have a diploid generation as well as they have a haploid generation. Okay, so let us see this image. Hmm. If you see this, let us start with gametes. So we all know that gametes are haploid in nature. Okay, so just a minute. Yeah, sorry. So gametes are haploid in nature. So here you can see that this is a haploid gamete 1n, 1n. After fertilization, it forms 2n. That is, now here it is diploid. This 1 and 1n were haploid, and after it forms diploid. And now this diploid will undergo mitosis. It will undergo several mitotic divisions, and it will form an organism which is 2n organism. That means the genetic content is diploid in here. Okay, but it can also skip this mitotic part, and it can undergo directly to meiosis. So this is one path, this is another path. Then here also you can see either this is 1n, either this 1n can undergo mitotic division and then it can enter for fertilization, or this 1n can directly come and start its fertilization process. So you have a diploid generation as well as a haploid generation when we talk about plant developmental biology. And both the generations are prominent generations. It's not like that the haploid is only seen in gametes. No, after even after gametes, you can see that your haploid generations are there. So this is a difference between plant and animal developmental biology basics. Then if we see here, so this is a figure. Let me just zoom it. Yeah. So this is a figure that shows both male and female reproductive systems of a plant. So if you see, this is a flower. So this carpel is what? It's the female reproductive organ, and this stamen is a male reproductive system. Now here you can see that this um, stamen, it consists of anther and filament. This carpel, it has ovaries which has ovules, and you have stamen in the branch, in the point. And this ovule is what we call as megasporangium, and microsporangium is what? The male sperms. Those are microsporangium. So you can just go through these images and you can just read it out and things would be even clearer to you. Okay, so now we'll start the questions. Fine. 1 minute. Okay. Okay, just a minute. Okay, so now we'll do the questions. So the first question is, identify A, B, and C in the following figure. So in this figure, we have to identify what this A, B, C, D refers or represents. So the options are A, synergids, B is polar nuclei, C is antipodal cells, and D is egg. Second is A, antipodal cells, B is polar nuclei, C is synergids, and D is egg. Third option is A is antipodal cells, B is egg, C is synergids, and D is polar nuclei. And the fourth option is A is synergids, B is egg, C is antipodal cells, and D is polar nuclei. So we have to identify what these A, B, C, D represents. So what it should be? Think. Think what it should be. Okay, let us see. So the correct answer is A is antipodal cells, B is polar nuclei, C is synergids, and D is egg. Now let us try to understand this question. Okay, so these are your antipodal cells. These are the A is your antipodal cells. Okay, these are the antipodal cells, and these are your polar nuclei. Okay, your these are synergids. Just a minute. and this is egg. Okay, so now what are antipodal cells and what are synergids? So antipodal cells are basically a structure which is present in flowering plants, okay, which provides nutrition. So these antipodal cells are required for nutrition. And these synergids basically guide the pollen tube towards egg during fertilization. Okay, so this is the egg. You can see the D option, it represents the egg, right? So these synergids, they actually guide the pollen tube towards the egg during the fertilization process. So what are these egg synergids? Synergids, they guide the pollen tube towards egg during fertilization. Okay, and these are your polar nuclei and these are your cells. So this is an easy question. Fine. Okay, next is a single ovary from a plant was isolated and subjected to chromosome banding. It was observed that out of the 7 cells within the embryo sac, only one cell contained a complete pair of homologous chromosomes, while the rest of the cells only had one of the two homologous chromosomes set. Based on this information, which of the following may have been the plants under observation? Options are A. Cycas, B. Pinus, C. Rose, D. Hibiscus, and E. Maize. So, what do you think should be the answer? Think. Just read the question carefully and try to think what can be the answer. Okay? Okay. I'll give you 1 minute to think and then I'll tell the answer. Fine. So now let us try to solve this question. Hmm. So the question here is referring to the presence of haploid and diploid cells within the embryo sac of the plant. Okay. Now, gymnosperms. In angiosperms are your flowering plants. So in angiosperms, such as what are the angiosperms or the flowering plants here? These are rose, hibiscus, and maize. So, in your angiosperms, which are your flowering plants, so the embryo sac consists of 7 cells, where the egg cell and other cells are haploid, and the only central cell that has two polar nuclei, which is diploid. Okay, so we are talking about angiosperms or the flowering plants. These are our flowering plants. So these flowering plants have such a kind of a system. You see here, this is nothing but a system, but this figure, this is actually your embryo sac. This is your for a flowering plant, where what you have? You have a 7 cell arrangement. So let us count what are the 7 cells. Your 1, 2, 3, 4, 5, 6 and 7. So you have a 7 cell arrangement. Okay, 7 to 8 cells you have, where all the cells are haploid. Your antipodal cells are also haploid, your synergid is also haploid, and your egg is also haploid. Or haploid. Only the central cell, it has two nucleus and it is diploid. So this kind of a structure or this kind of an arrangement is present only in your flowering plants, which are known as angiosperms. So here you can see that rose, hibiscus and maize, these are your angiosperms. So the question says that it was observed that out of the 7 cells within the embryo sac, only one cell contained a complete pair of homologous chromosomes, while the rest of the cells only had one of the two homologous chromosome set. What does it mean? That only one cell is diploid and rest of the cells are haploid. So this means they are referring to this kind of an arrangement where your central cell is diploid while rest all the cells, that is your antipodal cells, your synergids and your egg, your rest all of the cells are haploid. So based on this information, which of the following may have been the plants under observation? So from here we know that rose, hibiscus and maize, they are angiosperms. That means they will have such an arrangement. And what are cycas and pinus? Cycas and pinus are your gymnosperms. Okay, and they have a different type of a female gametophyte structure, and they do not follow this 7 cell or 8 nucleate arrangement which is found in angiosperms. So your angiosperm shows a 7-celled and 8 nucleate structure in the embryo sac. Now what is the meaning of 7-cell and 8 nucleate structure? Because there are 7 cells, but out of the 7 cells, 6 cells are haploid and 1 cell is diploid, the central cell. That means one cell has 2 nucleus. So overall how many nucleus are there? There are 8 nucleus. Although there are 7 cells, but there are 8 nucleus because the central cell is diploid. So your right answer would be rose, hibiscus and maize. Fine. Okay. Just give me 1 minute. Hmm. Now we come to the next question. So the correct answer is rose, hibiscus and maize. Fine. So the next question is, the total number of chromosomes in wheat is 42, that is 2n. Select the total number of chromosomes considering only the cells involved in pollen tube generation and fertilization. Your options are A 7, B 14, C 21, D 42, E 63 and F 84. So you have to select the total number of chromosomes that are involved in pollen tube generation and fertilization. So what do you think should be the answer of this question? Whatever you think, you just write it in the message box, okay? 2 minutes for this question. One minute left. Okay. So now let us see the question. The question says the total number of chromosomes in wheat is 42, that is 2n. Select the total number of chromosomes considering only the cells which are involved in pollen tube generation and fertilization. So we have to see only pollen tube generation and fertilization. So now what happens over here is that if you have studied about triple fusion phenomena, triple fusion, so there are three cells that are involved. Three cells involved. So what is this? First is your one sperm cell which fertilizes the egg. It contributes to 21 chromosomes. Okay. So we have hmm, hmm. So you have one sperm cell. It contributes to 21 chromosome. Then you have egg cell. One egg cell. It contributes to 21 chromosomes. And there, thus a second sperm, it fertilizes the central cell. Okay. So there is a second sperm also. It fertilizes the central cell. Fine. And here from here also you get 21 chromosomes. So now it is N, N, N. So total how much it would be? 3N. So which is 21 into 3, which is 63. So the correct answer is 63. Is it clear to everybody or not? So the question says select the total number of chromosomes considering the cells which are involved in pollen tube generation and fertilization. So in pollen tube generation fertilization overall two sperm cells are involved and one egg cell is involved. One sperm cell contributes to 21 chromosome. Second sperm cell, it fertilizes the central cell and again it is contributing to 21 chromosomes, and there is another one egg cell which is involved in fertilization. So you have 3n, that is 221 into 3, which equals to 63 chromosomes. 63 is the correct answer. Yes. So I hope this question is clear to you. So why we are dealing these questions? These kinds of questions will be asked in your assignments. So the questions that are already given in your assignments, if you have an idea by solving these questions, you can easily try to solve those questions as well. Okay. So we have to have an idea of how to solve these questions so that it's easier for us to solve the assignment questions. Fine. Okay. Now next question says, given below are the few statements regarding gametogenesis and fertilization in plants. Choose the incorrect statements. Okay. First statement is removal of septum will not affect the development of megaspores. Second, the haploid megaspore mother cell fuses with the generative nuclei to form the zygote. Third, in both megasporogenesis and microsporogenesis, the final division is meiotic that produces haploid gametes. Then, next, monocot endosperm forms by the fusion of a sperm cell with two polar nuclei. Then, dicot endosperm forms by the fusion of a sperm cell with two polar nuclei. And the last statement is excision of placenta will inhibit fertilization. And we have to choose the incorrect statement. Okay, not the correct one, the incorrect statement. So I'll give you 3 minutes to solve this question. And after 3 minutes, we'll discuss this. Okay. think think of all the options that are given. Okay, and why I give you time, because there should not be spoon feeding that I'm just telling you all the answers. You should also try to apply your brain and logic and try to think. One more minute left. Okay. Now let us try to see this question. So here we have to choose the incorrect statement first of all. Okay. So whenever you're solving any kind of multiple choice questions, always read the question carefully. For example, you just read that, okay, these are the few statements given. So by default, we think that we have to choose the correct statements. But no, sometimes the question can be tricky in the sense in language wise, and they can ask you to choose the incorrect statement. So this is one thing which you should always try to focus on. Okay. Now let us see all the questions. All the options. The first is removal of teptum will not affect the development of megaspores. For this, we should know what is teptum. So, teptum is the innermost layer of the anther wall, which provides nutrition and regulatory molecules to the developing pollen grains. Okay, so if I say, I just have this. If I ask what is teptum, okay, so this is the innermost layer of the anther wall. It's an innermost layer of the anther wall, and it provides nutrients to the developing pollen grain. Provides nutrients to the developing pollen grains. So from this, what can we infer? We infer that it is related to megaspores or microspores. First of all, you should know what is the difference between microspore and megaspore. Do you know the difference between megaspore and microscope? Can anybody tell me? So there are two things. First is microspore, okay, and the second one megaspore. So your microspore is nothing but your male sporangia, that is the male reproductive of organ. Male sporangia. And your megaspore is your female sporangia. So micro, why? Because your pollen grains are smaller than your ovum or the egg. So that is why it is a micro, and here it is mega. So microspore is nothing but your male sporangia, that means your male reproductive organ, and megaspore is your female reproductive system organ. So now the question here says removal of teptum will not affect the development of megaspores. So is this a correct statement or incorrect statement? Megaspore means ovum. So is it correct? Yes, it is correct. Why it is correct? Because we have just studied that septum, basically it's a layer of the anther wall, and it provides nutrition to the developing pollen grains. So this means that it is related to microsporangia and not the megasporangia. That means it is related to male reproductive organ, not the female one. And megaspore is the micro, means your female organ. So this statement is correct. It will not affect the development of megaspores. It will affect the development of pollen grains, microspores. Okay. So this is your correct statement. Then next is the haploid megaspore mother cell fuses with the generative nucleus to form the zygote. What does this mean? Is this a correct statement or incorrect statement? It is an incorrect statement. Why? Because first thing here it is written is the haploid megaspore mother cell. Your mother cell is haploid or diploid? Your mother cell is diploid in nature. So your megaspore mother cell is diploid. So the megaspore mother cell is diploid and it undergoes meiosis to form megaspores. So megaspores are formed from megaspore mother cell. Okay, so your megaspore mother cell is diploid in nature and basically it undergoes meiotic division to form your microspore, sorry, megaspores. So megaspores are your haploid in nature then. So both megaspores and microspores, that means both your egg and your pollen grains, they both are haploid in nature. But your ovum, I mean the egg, it is formed from the megaspore mother cell, and these megaspore mother cells, they undergo the meiotic division to form megaspores. And the egg, which is haploid, it fuses with the sperm nucleus to form the generative nucleus, which ultimately forms zygote. So here it is written that the haploid megaspore mother cells fuses with the generative nucleus to form the zygote. So first of all, your megaspore mother cell is not haploid, it is diploid. Second, these megaspore mother cells are not fusing with the is not fusing with the generative nucleus, but your haploid sperm fuses with, or haploid sperm nucleus, it fuses with your haploid egg to form the zygote. Okay, is it clear? Fine. This is your incorrect statement. Your next statement is in both megasporogenesis and microsporogenesis, the final division is meiotic that produces haploid gametes. Now, this is again an incorrect statement. Why? Because the final product of megasporogenesis and microsporogenesis are not gametes, these are spores. The gametes are produced later through mitosis in the female and male gametophyte. You understand? So, your megasporogenesis and microsporogenesis do not produce gamete. They produce spores. Your gametes are your egg and sperm. And these are formed later through the mitosis which occurs in the male and female gametophytes respectively. So, this statement again becomes incorrect. Then your next statement is monocot endosperm forms by the fusion of a sperm cell with two polar nuclei. Is it a correct statement or an incorrect statement? It is correct. So, we know that in monocots, what happens? Double fertilization happens. So, during double fertilization, sperm cells, it fuses with two polar nuclei. So, this is a correct statement. Then dicot endosperm forms by the fusion of a sperm cell with two polar nuclei. Yes, it is also correct. Similar to monocots, dicots also form endosperm by the fusion of a sperm cell with two polar nuclei. So, this is again correct statement. Then excision of placenta will inhibit fertilization. Now, this is a general statement. I mean, not general, but it's a general knowledge thing that you should know that Placenta, it supports, provides the nutrient, it supports the ovule, and obviously its removal would affect the fertilization process. So from this, what we can say, what are the incorrect options? There are two incorrect options. First is, in the haploid megaspore, mother cell fuses with the generative nucleus to form the zygote. And the second one is, in both megasporogenesis and microsporogenesis, the final division is meiotic that produces haploid gametes. So these are your two incorrect statements. So let us see this. So I was talking about tapetum. So what is tapetum? Basically, it is the innermost layer of the anther wall. So just if you cut an anther cross sectionally, this is a cross sectional, so you can see it like this, and the innermost layer is your tapetum. Okay, you can see this layer is your tapetum, which provides what? Nutrition to the pollen grains. Fine. Now we'll come to the next question. The next question is, inability of the gametes from the same parent plant to fuse and form the zygote is known as A. Sporophytic self incompatibility only, B. Gametophytic self incompatibility only, C. Either gametophytic or sporophytic self incompatibility, and D option is the last option, interspecific incompatibility. What is the correct answer? You have to tell. So 2 minutes, and then I'll switch and I will tell the answer. Hmm. Think. 1 minute left. Hmm. Over. Fine. Okay. Now let us try to solve this question. So now what is this inability of the gametes from the same parent to fuse? This is known as self incompatibility. The inability of gametes from the same parent to fuse and form a zygote is known as self incompatibility. Now, this self incompatibility can be of two types. First is sporophytic self incompatibility, and the second one is gametophytic self incompatibility. Now, sporophytic self incompatibility occurs when the incompatibility is determined by the diploid genotype of the sporophyte, that is, the pollen producing plant. So, there is self incompatibility. If we talk about self incompatibility, okay, this can be of two types. First is sporophytic, and the second one is gametophytic. Okay, this sporophytic self incompatibility is determined by the plants that produces pollen. That means plant that produces pollen. And this gametophytic self incompatibility is determined by the haploid genotype of the pollen itself. So this sporophytic self incompatibility is determined by the diploid genotype of the sporophyte. Remember, diploid genotype of the sporophyte. This sporophytic self incompatibility, diploid genotype of the sporophyte, whereas this gametophytic self incompatibility is determined by the haploid genotype of the pollen itself. Haploid genotype of the pollen itself. So there can be two kinds of self incompatibility, either gametophytic self incompatibility or sporophytic self incompatibility. Fine. Now, next question is, sperm cells show chemotatic movement towards A. Potassium waves, B. Calcium waves, C. Chloride ion waves, or hydronium or hydrogen ion waves. Hydrogen ion waves. So, what is the correct option? 2 minutes and then we'll see what is the right answer and the explanation as well. Hmm. 1 more minute left. Think. Hmm. Okay. So, what should be the correct answer here? The correct answer is calcium wave. So, calcium ions, they play a key role in the signaling pathways that guide the sperm cells toward egg, especially during the fertilization. So, this chemotactic movement is shown by the calcium waves. Why? Because these calcium ions, they play an important role which guide the sperm cells towards the egg. So, calcium ions, they basically guides the sperm cell towards the egg especially during fertilization. Okay. So why I write here is because if my voice is not clear due to some internet connectivity or some issues, at least if I am writing it, you're able to read it and understand. Okay. That's why I try to write over here, because we are doing these classes online. So we don't really know that our connection is proper or not. Sometimes from my end, the connection is not proper. Sometimes it can happen that connection is not proper from your end. So that's why I tend to write things. So I hope questions up till now are fine. In between, we'll also see some of the theory part, because theory part is also very important when we consider some new topics, such as especially like plant developmental biology. Okay, so after this question, we'll just go through a little bit theory. So the question says apical basal axis of the embryo is determined by first dash but interrupted by dash. Okay, the options are direction of light and gravity, and second is position of egg, point of sperm entry or direction of light and gravity, position of nuclei or direction of light and gravity, and the last option is position of egg and calcium level. So 2 minutes and then I'll switch the screen. It's 6:55, 6:57. We'll try to solve it. One minute more. Think. Okay. Few seconds left. Fine. Now let us see. The question says apical basal axis of the embryo is determined by what? So, this apical basis axis development, it begins right after the fertilization, and it involves the establishment of polarity in the plant embryo. So this apical basis axis basically helps in the establishment of polarity in the plant embryo. Now, the point of sperm entry, which is your fertilization, it determines or it helps in determining the initial orientation of the apical basal axis. So first we'll see what is the correct option. The correct answer is point of sperm entry, and second is direction of light and gravity. That means this one. This one is the correct option. That means apical basal axis of the embryo is determined by point of sperm entry, but direction of light and gravity interrupts it. Interrupted by direction of light and gravity. So now establishment of polarity in the plant embryo is determined by apical basis axis. So the first thing that directs the apical basal axis formation is the point of sperm entry. That means the point where fertilization has taken place, that determines the polarity. But environmental factors such as light and gravity, it can influence and it can further redirect or shape the development of the axis. But the initial establishment is always created by the point of sperm entry. Okay, so the initial polarity formation or the initial axis formation is determined by the point of sperm entry, but these are the environmental factors such as light and gravity. These are your environmental factors, and these environmental factor at the later stages, it can interrupt uh, the axis formation, and it can um, further change the development of the axis. Fine. So this is option number 2 is the correct answer. That is point of sperm entry and direction of light and gravity. Now let us quickly see. So you can see here. So I don't know how many of you are from the plant background. So students who are from the plant background, it is easier for them because they know what is an anther, what is a filament, what is stamen, what is the female reproductive system, everything. But students who are coming from animal background, they need to have little bit of basic understanding of the the basic terminologies which are used in plant developmental biology, and the basic structures and very basic things about plant developmental biology, the structures, the naming, the terminologies. So if you are not very much familiar with it, I would suggest you just go through some of the basic textbooks and just get an idea. Even Gilbert's is fine. You can have an idea of the plant system, developmental biology system in Gilbert's as well. And you can just either you can just Google it, just have a little bit basic idea about how this uh, the morphology of the male sporophyte and female sporophyte, or the male gamete and female gamete is. So this is your stamen, which has anther and filament, and this is a cross sectional area. If you just cut it like this cross sectional area shows the pollen sacs. Fine. Then so pollen mother cell or microspore mother cell. So how it happens in the case of monocots, how it happens in the case of dicots, you can see. You just I'm just giving you a brief. And this is your female megasporangium. So your microsporangium, you see here. See, these are microspores or mother cells. But in the case of females, you have only one mother cell. Okay, you only have one megaspore mother cell. Unlike in the microsporangium where there are numerous, in the case of megasporangium, you have only one mother cell, megaspore mother cell. This is sigma, this is style, ovule, and this is the ovary wall. And this connection that you see, this is your placenta. This is your megasporangium where the actual fertilization takes place, and these three are in the later stages, these are degraded. And then this is the figure. You can see here, these are your antipodal cells. This is the polar nuclei, and these are your synergids. These are two synergids. See, This is the one that has been asked, the question. These are your two synergids. This is your egg. These are the polar nucleus. There are two polar nucleus and these are your antipodal cells. This is your micropyle. Just to see how the structure looks. Now, you can have two different types of plants. You can have dioecious plants or monoecious plants. So, this refers to the landing of pollen on stigma and germination of the pollen. So, monoecious plants means they form both type of gametes. That means male and female gametes are produced by the same plant. Monoecious plants, they produce both male and female gametes. Now, what is the difference here? You can have two kinds of flowers: perfect flower, carpellate flower, and staminate flower. What are perfect flowers? That means in the same flower, both stamen and carpel are present. That means both male and female gametes are present on the same flower. Carpellate, carpellate flower means plant is same, but one flower will have only carpel, another flower will have only stamen. For example, maize. So, staminate flower means which has only stamen. That means either you have a perfect flower, that means in the same flower you have both male and female gametes. That means you both have same flower has stamen and carpel both. Or you can have that in a plant there are numerous flower. In one flower you have only carpels, in other flower you have only stamen. So the plant is same, but here the flowers are different. So the flower which has only carpels are known as carpellate flower. The flower which has only stamen, they are known as staminate flowers. But all this comes under the category of monoecious plants. Then dioecious. What are dioecious plants? These are the plants where carpel and stamen are present on separate plants. Okay, so carpellate and staminate flowers are produced on separate plants, whereas in monoecious, carpellate and flowerate are produced in the same plants. And there can be in the same plant, the same flower or different flower, that can differ. So, for example, some kinds of palm trees, not all kinds of palm trees, but some kind of palm trees are dioecious plants. So, you see here, first we'll see the example of monoecious flower, perfect flower. You see hibiscus. So stamen and carpel, this is your carpel and this is your stamen. So stamen and carpel both are present in the same flower. This is your hibiscus plant. Okay. This is your hibiscus plant. And here you see maize, female and male flowers are separate but on the same plant. This is important that in maize, female and male flowers are separate, but they are present on the same plant. Okay, you see in the same plant, see here, just a minute. Yeah, in the same flower, some are have the carpel, while some has the stamen. Okay, and these are the palm trees. So, palm trees, male and female gametes are present on the different plants. Okay, so these are your examples. Now we'll come back to the questions. Hmm. Okay, we did this question. We had no, oh, sorry, I just showed you the answer. Anyways, now the question says the absorptive surface of the embryo is oriented towards dash by dash. The options are soil and basal cell, light source and basal cell, water source and suspensor, and food source and suspensor. So now you have to tell the correct answer, the correct option from this. 2 minutes, then we'll discuss. Okay. It's 7, 8. At 7:10, we'll discuss the question. Think. 1 more minute left. Hmm. Okay. Okay. Let us see. Okay. So, what is the meaning of the absorptive surface of the embryo first? So the absorptive surface of the embryo, it refers to the part of the embryo that is specialized for taking nutrients and water from its surroundings. Okay, this is a part of the embryo that is specialized for taking nutrients and water from its surroundings. So, in plant embryo, this surface often develops in relation to structures that provides essential resources, such as the endosperm or the suspensor, which connects the embryo to nutrient sources in the seed. And this absorption is crucial for the embryo's growth and development because it establishes itself before becoming photosynthetically active. So, in that context, the correct answer would be food source and suspensor. So, suspensor is a region of a plant embryo that connects to the surrounding tissue and it helps to develop. So, the answer becomes the absorptive surface of the embryo is oriented towards the food source by the suspensor. So this why it is oriented towards the food source, because this absorptive surface it it is oriented towards the area which from where it can take the nutrients and water from the surrounding. So whatever the surrounding which has the food source, in that orientation only this absorptive surface of the embryo would be oriented with the help of the suspensor. So the correct answer is last one. Okay. Now the next question is, pin localization is mediated by pin protein kinase, PID protein, PID protein kinase, PDI protease, or pin phosphatase. Okay. So what is the correct answer? 1 minute. It's a simple question, so I'll just give you 1 minute to solve this question. Hmm. Okay. So 1 minute is over. Now, what is the meaning of pin localization? Pin localization in context of plant developmental biology refers to the spatial arrangement of pin proteins which are involved in the transport of a key plant hormone which is auxin. Okay. So pin localization is the spatial arrangement of pin proteins, and these pin proteins, these pin proteins, these are involved in the transport of auxin. These are involved in the transport of auxin. Now, these proteins, that means these pin proteins, they are located in the plasma membrane of the plant cell, and it play a crucial role in directing the flow of auxins. So it influences processes such as cell elongation, tissue patterning, and organ development. So these pin proteins, what do they do? These pin proteins, they have an important role in directing the flow of auxin hormone. So they help in directing the flow of auxin. Okay. So this is about pin proteins and its localization. So they are localized where? They are localized in the plasma membrane of the plant cells. Now, how their localization is carried out? Their localization is helped with the help of protein kinase. But which protein kinase? Pin protein kinase or PID protein kinase? The answer is PID protein kinase. So the correct answer is C. So these PID is pin formed interactory kinase. Pin, pin formed interactory kinases. It helps in positioning the pin proteins correctly within the cell. So up till now we understood that what are pin proteins. So pin proteins are nothing but they are help in the transport of auxin, and these pin proteins direct the flow of auxins, and ultimately by directing the flow of auxins they influence processes such as cell elongation, tissue patterning, and organ development. Now these pin proteins localization is mediated through PID protein kinase, which are pin formed interactor kinases. These PID kinase or PID protein kinases, they help in positioning the pin proteins correctly within the cell. Okay. So just I'm writing it for your own convenience. Okay. So PID, which is your pin formed interactor kinases, they help in positioning the pin proteins. They help in positioning the pin proteins. Now, important point to remember here is that the localization of the spin protein is often polar. What does it mean that the localization is polar? Means that they are concentrated on one side of the cell. Now, why you think that this polarity has to be maintained? This polarity has to be maintained because it allows for the directional transport of auxins, which is essential in establishing gradients that guides the growth and development. So, this is just an important point to know that this pin localization is mediated by PID protein kinase, and the pin localization is often polar, which means that they are concentrated on one side of the cells, and this polarity allows for the directional support of auxin, which is essential for establishing gradients that guides its growth and development. So I think this is clear to you. So whenever we try to solve a question, we try to not only just mark the correct answer, no. You should also try to get the concept, the theory behind the question. Just by looking at the question that pin localization is mediated by PID protein kinase, no. You should know the before and after of the question. You should know the background of the topic, because like this only you will get through questions. We can complete the whole topic actually just by solving the question and reading a little bit background of it. So that is why my focus is that whatever questions that we are solving, we should know the concept behind those questions, and we should understand the concept that that question is talking about, because that is the most important thing. Otherwise you can just solve 100 of questions, but if you do not gain anything, then it's a waste. Fine. Now next question is, DCL codes for A, DROSA, B, Dicer ortholog, C, SUS1, and D is DGCR. So DROSA, Dicer ortholog, SUS1 or DGCR. I'll give you 2 minutes to solve this question and then I will switch the screen, okay? It's 7:21 and 7:23 will switch. Think. One more minute left. One 30 seconds left. Okay. So the correct answer is Dicer ortholog. So DCL1, the full form itself says dicer like one. Okay. So DCL1, the full form is dicer like one. So basically DCL1 is an enzyme which is found in plants, which is important for the biogenesis of microRNAs and small interfering, that is siRNAs. and it functions by processing precursor RNA molecules into shorter double-stranded RNA fragments which are involved in gene regulation. And this DCL1 specifically plays a key role in microRNA pathway where it helps to produce mature RNA from the pre-mRNA. Now, these miRNAs are important for regulating gene expression. How? Because they guide silencing compound the mRNAs, which ultimately helps in the degradation or inhibition. So, if you remember, just I'm telling you, in normal, if you have studied about the normal miRNA biogenesis, you know that there is primary miRNA, pri-miRNA. Okay, this primary mRNA is converted into pre-miRNA, and this is mediated by DROSHA. And this pre-miRNA is in the nucleus. Now, from the nucleus, it will come out to the cytoplasm. So it will come out of the nucleus. It will come out of the nucleus, and then what will happen? Then again, it is processed by dicer in the cytoplasm, and then it forms a mature strand, mature miRNA, and a passenger strand. And this mature strand ultimately helps in miRNA based inhibition. So this DCL1, it is basically your dicer ortholog, and its full form is dicer like 1. Okay. Next question is, which of the following statements regarding embryonic development of plants are correct? First is, first embryonic division is asymmetric. B, in some species, the root meristem is partially derived from the hypophysis. C, axial polarity is first distinguished at the globular stage. D, apical meristem is involved only in shoot development. And E, supporting structure and the shoot form first, followed by the light capturing leaves, which is followed by the roots. So I'll give you 3 minutes. You think of all the options. It's 26 right now, 7:26. 7:29 we'll discuss it. Okay. Just read all the options, and important is that all the options are not connected. These are different statements. These are not the statements of a one concept. So all the statements are basically different concepts. Okay. Just read all the options and think what can be the correct answer. What are the correct statements? 1 more minute left. It's 7:28 now. 30 seconds left. Okay. Now let us see. So we'll one by one we'll try to see understand each line, and we'll try to find out whether these are correct statements or not. So the first statement is first embryonic cell division is asymmetric. So is this a correct statement or not? This is a correct statement. So the first division of a zygote is typically asymmetric, which leads to different sizes and fates for the daughter cells. So the first embryonic cell division is asymmetric. Then, in some species, the root meristem is partially derived from the hypophysis. Is this a correct statement? Yes, it is also correct. So, the hypophysis is the basal cell and it contributes to the formation of root meristem in many plant species. Then, third is axial polarity is first distinguished at the globular state. Yes, is it correct or not? Hmm? Axial polarity is first distinguished at the globular state. No, it is incorrect statement. Axial polarity is typically established earlier during the zygotic stage or at the proembryo stage, but not at the globular stage because your axial polarity is established earlier. Then D is apical meristem is involved only in shoot development. It is again incorrect. Sorry, this is incorrect. So it is incorrect. Why? Because the apical meristem contributes to both shoot and root development, not only to shoot development. So this apical meristem, it contributes to both shoot and root development, although there are distinct type of meristems for each, but apical meristem is involved in both shoot and root development. Then E is supporting structure and the shoot form first, followed by the light capturing leaves, which is followed by roots. This is also incorrect. Why? Because while shoots do develop early, roots typically start to form alongside or shortly after the shoot rather than being developed last. See, here it is written supporting structures in the shoot form first. Yes, shoot forms first, which is, but it is not like that roots are formed at the last. No. So, roots are also more or less they are formed at the same time. Little bit delay can be there, but it is not like that roots are formed at the last point. No. So this becomes again your incorrect statement. So your correct statement is first embryonic division is cell division is asymmetric, and in some species the root meristem is partially derived from the hypophysis. These are your two correct statements according to me. Let us see. Yes. So the correct answer is this only. Okay. Okay. Now the next question is which of the following statements regarding auxin signaling are correct? An inhibitor of tryptophan synthase adversely affect auxin mediated expression. B. Cells with a higher concentration of spin carriers will have a low concentration of auxin. C. There is a reversal in polarity of auxin transport from the suspensor to the embryo that promotes root development. D. Auxin dependent genes are highly expressed in the suspensor. And E. PP2A inhibition will increase auxin efflux from the cells. So you have to tell which of the following statements regarding auxin signaling is correct. So these all the statements they talk about the auxin signaling. So for this you need to have an idea of how auxin signaling works. So I'll give you 3 minutes to solve this question. It is 7:33 right now, and we'll discuss this question at 7:36. Okay? Try to understand all the options. So in the last question, all the options were unrelated to each other, but these all are related to auxin signaling only. Okay. 1 more minute left. Okay. So let us see. The first option says, stop a minute. Hmm. An inhibitor of tryptophan synthase adversely affects auxin mediated expression. So, will this be a correct statement or an incorrect statement? This will be a correct statement. Why? Because what is the precursor of auxin? Hmm? What is the precursor of auxin? So, auxin is formed from tryptophan. So tryptophan is the precursor for auxin. So tryptophan synthase is what? An enzyme which produces tryptophan. If tryptophan synthase is inhibited, then tryptophan will not be formed. If tryptophan is not formed, then your auxin signaling is inhibited because auxin is not formed. If auxin is not formed, then auxin mediated expression is also not formed. So an inhibitor of tryptophan synthase adversely affects auxin mediated expression. It is correct because tryptophan is the precursor of auxin and tryptophan synthase synthesizes tryptophan. Okay. The next option is cells with a higher concentration of pin carriers will have a lower concentration of auxin. Now, again, higher concentration of pin carrier will have a lower concentration of auxin. That means pin and auxin concentration are opposite to each other. If pin is more, auxin is less. If auxin is more, pin is less. So, is it a correct or an incorrect statement? So, this is a correct statement. Why? Because when there is higher pin expression, then there is more auxin transportation out of the cell. So, which reduces your local auxin concentration. So, higher pin production means higher pin expression. Higher pin expression means that more greater auxin transport out of the cell. Greater auxin transport out of the cell. So therefore the local auxin concentration it reduces. Therefore B is also correct, that is cells with a higher concentration of pin carriers will have a lower concentration of auxin. Then next is there is a reversal in polarity of auxin transport from the suspensor to the embryo that promotes your root development. This is incorrect statement. Why? This is not a universally correct statement, not a completely correct statement, because the polarity changes, it is developmental stage specific. So polarity changes depend on specific developmental stages. So this is not a completely correct statement. So this is wrong. Next, auxin dependent genes are highly expressed in the suspensor. This is correct. The suspensor does not express auxin related genes, therefore it contributes to signaling. Sorry, suspensor, it expresses. Yeah. So the suspensor, it expresses auxin related genes which helps in the signaling. Then PP2A inhibition will increase auxin efflux from the cells. It is also correct statement. So PP2A inhibition. So when your PP2A is inhibited, then auxin is effluxed from the cells. That means oxygen is taken out from the cells. There are two terms, influx and efflux. Influx means inside movement and efflux means outside movement. So when there is an inhibition of the PP2A enzyme, then what will happen? That auxin is taken out from the cell. Why it is taken out from the cell? Because the transport proteins are changed in such a way that your auxin is taken out from the cell. So this is a correct statement. So inhibiting PP2A can enhance the auxin transport by affecting the regulation of transport proteins. Okay, fine. Now next question. So yes, so the accepted answers are A, B, D and E. See, an inhibitor of tryptophan synthase adversely affects auxin mediated expression. Cells with a higher concentration of pin carriers will have a lower concentration of auxin. Auxin dependent genes are highly expressed in the suspensor, and PP2A inhibition will increase auxin efflux from the cells. Okay, next question. Which of the following statements regarding axis development are correct? Now here it's this question is not a plant question, it's an animal uh, Drosophila development a question, but because we haven't covered this particular uh, topics question, that is why this there's one question that I've included here. Okay, so the options are, so here you have to select the correct statement regarding axis development, specifically with reference to Drosophila, because that is what we have learned till now. So your first option is the homeotic genes that specify the anterior posterior order of organs are arranged 5 to 3 prime on the chromosome. B. Mutation of a homeotic gene will result in transformation of structures formed. C. Mutation in the cis regulatory regions of homeotic genes will shift the position of a given organ. D. Once specified, these genes are acetylated or methylated to continue a stable pattern of expression. And E. Loss of function mutation in pair rule genes shift organ boundaries. So you have to select the correct options among all the given options, and there are, I will give you 3 minutes. It's 9:43. We'll switch the screen at 9:46. Read all the options. You must have studied about homeotic genes. We have also discussed this topic a lot in the class. And in your this week's lecture also there was again some portion about homeotic genes. Try to solve the question. Okay, 30 seconds left. Okay. Now let us try to understand all the options. The first option is the homeotic genes that specify the anterior posterior order of organs are arranged 5 prime to 3 prime on the chromosome. Is this a correct option? Yes, it is correct. Hmm? No, it is not correct. See, the homeotic genes that specify the anterior posterior order of the organs are arranged 5 prime to 3 prime on the chromosomes. It is the homeotic genes, they are arranged in a linear order, okay, and these linear order corresponds to their expression along the anterior posterior axis, but that is the case in drosophila. But since here it is not mentioned about drosophila, therefore in that accordance we will say it's wrong. Otherwise for drosophila it is right. Then mutation of a homeotic gene will result in transformation of structures formed. Is this a correct statement or not? It is a correct statement. Why it is a correct statement? Think, what is the function of homeotic genes? First of all, tell me what are homeotic genes and what is their function? Okay. See, homeotic genes are regulatory genes which controls the development of anatomical structures in your animals, also in plants they are present. So basically, if there is any kind of mutation in homeotic genes, it can lead to homeotic malformations or homeotic transformations. What kind of transformations? For example, your antenna is replaced with the legs, your antenna is transformed into legs, or there is ectopic eye expression on wings, legs, and antenna. Therefore, if there is any mutation in the homeotic genes, it will result in different structure formation. The place where antenna should be formed, their legs are forming. The place where eyes are formed, their antenna is forming. So these kinds of mutations can just lead to a malformed body. So this is your correct statement. Your next statement is mutations in the cis regulatory regions of a homeotic gene will shift the position of the organ. So can it happen? Is it a correct statement or not? Yes. Why? Because when there is a mutation in the cis regulatory elements, then there is a change in the expression pattern of homeotic gene. And when there is a change in the expression pattern of the homeotic genes, then there can be alterations in position and identity of structures. So, mutations in cis regulatory regions of homeotic genes will shift the position of the organism, true, because it can change the expression pattern of homeotic genes. Okay. So this is the correct statement. Then once specified, these genes are acetylated or methylated to continue a stable pattern of expression. Is it correct? Yes. So why? Because epigenetic modifications such as acetylation or methylation, they help in maintaining stable gene expression. So see, this is very important to understand. So whenever we talk about modifications, acetylation and methylation, they are nothing but epigenetic modifications. So what are epigenetic modification? Always remember that all the genes are present in all the cells and all the cells have the same genes. But the expression pattern differs from cell to cell. And this difference of expression pattern leads to differential gene expression in various cell types. So, for example, if you consider humans, genes which are present for which are specific for eye color, they're also present in kidney. Genes for some important function of kidney are also present in our eyes, but the expression of those genes decides which gene has to be active or not. So the gene which is responsible for eye color is not expressed in your kidney. So there is a differential expression pattern. Now, how this differential expression pattern is maintained? This differential expression pattern is maintained with the help of certain processes such as acetylation and methylation. Acetylation and methylation essentially means addition of acetyl groups or methyl groups, but the ultimate result of acetylation and methylation are completely opposite to each other. Just a minute. How they are completely opposite to each other? Because acetylation will cause the expression. Acetylation, it promotes expression, whereas methylation, it represses the expression. So acetylation promotes the expression, whereas methylation, it represses the expression. Okay. So the option says once specified, these genes are acetylated or methylated to continue a stable expression pattern. Yes, because these epigenetic modifications, acetylation or methylation, they help in promoting the stable pattern of gene expression. Then loss of function mutation in pair rule genes will shift organ boundary. Yes. So pair rule genes are crucial for defining segments in early development, and their loss can lead to shift or disruption of organ boundaries. So we have gap genes, we have pair rule genes. So these pair rule genes are what? They divide the body into segments. And when we are dividing the body into segment means that we are putting boundaries. That okay, acha, up till this region there would be head, up till this region there would be thorax, up till this region there would be abdomen, up till this region there would be tail. So if there is any mutation in the pair rule genes, then this boundary can be either increased or decreased. Your head can go up till from the anterior to the posterior end, or tail can be shifted to the anterior side. Anything can happen. So this is also a correct statement that loss of function mutation in pair rule genes affect the organ boundaries. Okay. Now we'll come to the next question. A pistil with S1 S3 genotype was pollinated by two pollens collected from plants of the same species, and SRK activation was recorded. Based on your understanding, select the correct inferences. A. Pollen A may have S1 allele. B. Pollen B may have S3 allele. C. Fertilization will occur in both cases. D. Fertilization will occur only via pollen B, and sporophyte of pollen A may have had S2 allele, and sporophyte of pollen A may have had S4 allele. Okay, so there is a graph. You can see here there is a graph. There, according to this graph, certain statements are written that you have to solve. So I will give you 3 minutes to solve this question and then we'll discuss it. Okay, it's 7:55. So we'll start. 7:58, we'll discuss this question, right? Think. Think. I just 2 minutes left. Hmm. 1 more minute and then we will discuss this question. Okay. It might take a few more minutes, but we'll try to solve this question because it is a very interesting question. And just for your clue, it is based on self incompatibility concept that we have already studied. So, because we have already studied this concept, that's why I'm trying to solve, at least cover this question in today's class. Okay. So, 1 more minute left, and then we'll try to understand the question. Okay, fine. Let us try to understand this. Now, the question says a pistol with genotype S1 S3 was pollinated by two pollens collected from plants of the same species and SRK activation was recorded. Based on your understanding, select the correct inference. First of all, we should try to understand what the question says. The pistol has a genotype of S1 S3. What does it mean? It means that it carries two alleles for self incompatibility. This is SI actually. Okay, self incompatibility in plant species. So these are the two alleles of self incompatibility. S1 and S3 are the two alleles of self incompatibility. Okay. Now, the two pollens likely from plants with different S alleles were used for pollination, and SRK, that means S receptor kinase activation, was observed. That means, so a female reproductive organ pistil was pollinated by two pollens which are collected from plants of the same species. That means these pollens are from the same species, and the genotype of the pistil is S1 S3, and then SRK activation was recorded. So SRK activation is a key step in self incompatibility. That means if there is a high SRK activation, that means it indicates a self incompatibility response. That means they are not compatible with each other. So SRK activation is a key step in self incompatibility, where the interaction between the pistil and pollen, it determines if fertilization will be blocked. Now, the correct answers are: pollen B may have S3 allele, sporophyte of pollen A may have S2 allele, and sporophyte of pollen A may have S4 allele. C. These are the levels of SRK inactivation. In graph A, you can say that in pollen A, SRK activation is less. In pollen B, through pollen B, SRK activation is more. So, when there is higher self incompatibility, then SRK inactivation would be even more. So, when there is more of self incompatibility, then there is more of SRK activation. And when there would be self incompatibility, since the genotype of the female reproductive organ, the pistil, is S1 S3, if S1 or S3 genotype is carried by the pollens, then there would be self incompatibility. Okay, now here you can say that in pollen A, the since the SRK activation is very less, therefore the self incompatibility is also very less. That means that pollen A do not have S1 or S3 genotype. But here you can see that in pollen B, the self incompatibility is very high because SRK activation is also very high. Therefore, it is likely that the genotype of pollen B is S1 and S3. So pollen A may have S1 allele? No, because the SRK activation is very less, which means self incompatibility is very less, which means that the genotype of the pollen A cannot be S1. Pollen B may have S3 allele, highly likely that pollen B can have S3 allele, because if pollen B has S3 allele and the pistil also has S3 allele, self incompatibility is very high, therefore SRK activation is very high, as we can see from the graph. Fertilization will occur in both the cases. Here, the chances of fertilization are very less because there is self incompatibility. So, this is wrong. Fertilization will only occur via pollen B. See, fertilization, because there would be some amount of fertilization in pollen A also, with pollen A also, but it would be very less. So, we cannot say it only occurs with pollen B. We can say that more will occur with pollen B. This is also wrong. Sporophyte of pollen A may have had S2 allele. Yes, because S see, the genotype of the pistil is S1 and S3. If either S1 or S3 is the genotype of the pollen, then there would be very much self incompatibility and SRK activation would be very high. But if the genotype of the pollen is different from S1 and S3, then there would be very less SRK activation, which indicates that self incompatibility is very less. So, pollen A can have a genotype of, may have S2 allele or S4 allele. It can have, because any other any allele other than S1 and S3 can be present in pollen A. So, these two are the correct statements. So your correct statements are B, E, and F. It is clear. It is so the time is up now. So only one question was left, I think. Yeah, only one question is left. But what is the question? But since it's a graphical question, I do not want it you to I don't want this question to be completed in hurriedly. So one question is left. This question we'll cover in the next class. The rest all questions were covered, and I hope this topic was clear to you. It's plant, so it is a little difficult uh, for the animal people to understand. But what I would suggest is you go through the books or Google it and at least clear your basics, because that is what is going to help you to understand this topic. Okay. So with this, we end today's class. Thank you, everybody.